Hi everyone, we are now in Module 3 and we're going to go over the lab kit labs that you have to do for Module 3. This module is all about metabolism and different reactions like anabolic and catabolic reactions and redox reactions that eventually lead you to understanding about cellular respiration and photosynthesis. So that's kind of what's covered in the readings. So you're definitely going to want to do the readings first, because those will definitely help you, as well as the videos that I posted in the class announcements. But going ahead and going to our lab kit labs, you'll see that there are three. Two of them are due at one time, and then the the third one is due at a different time. So we're going to go ahead and just quickly look at them. So this lab is about looking at cellular differences between eukaryotes, prokaryotes, and viruses. You're going to, as usual, have the overview and the background. So again, these activities take longer than the Module 2 labs did, on average about an hour to an hour and a half. This gives you the overview of cell transportation, so osmosis, diffusion, the difference between hypertonic, hypotonic, isotonic. There's really good videos in the class announcements if you are more of a visual person. And then it goes into the different types of cells and how they kind of evolved along the phylogenetic tree. So there's a lot of good background information in here. So you have this table here that tells you about the different components of the cell and its function, and whether it's in a prokaryote or a eukaryote. Prokaryotes don't have a nucleus, whereas eukaryotes do. Eukaryotes are definitely more complex than prokaryotic cells are. So you have materials in the kit, but then also pay really close attention to what you need to pick up. So you need 2 medium white potatoes from the store, some spoons, some paper, knife, a cutting board. Most of these things should be around your house, but you will have to go out and buy the potatoes. So in this first activity, you are going to be using the potato, and you're also going to be looking at, the surface area of cells. You're going to have the potatoes soak in a solution, and then you're going to observe, what happens after the soaking. So you let them soak for about 80 minutes. And then you're just going to make some cuts and make some observations measuring the size and the cross section of those pieces. For activity 2, this is where you are becoming familiar with how solutes and solvents kind of move across the cell membrane. You're going to have different amounts of sucrose in a cup and some in a dialysis tube, and the different concentrations should cause the water to move in and out of these tubing. So you'll take some time to allow that to happen, and then you're going to see, okay, did the dialysis tubing represents the cell. So you're just going to see, okay, did it shrink? Did it absorb more water? Like what happened based on the different concentrations. And then activity 3 is going to be looking at the different cell types. You're going to be drawing, and don't worry about if you're like, I'm not a good artist. I'm not either. So just do the best you can, and it'll be okay. So then you're going to actually design, based on what you know about eukaryotes and viruses, you're actually going to design one. So again, lab 3 is all kind of about drawing. These are your dichotomous keys that you're going to use in this lab. And these are kind of like a yes-no question. So for instance, If the first question is, right, does the structure lack internal organelles or it has internal organelles? So if it lacks, then you go to the virus. If it has internal organelles, then you just go to question 2. And then you just keep going with these directions, and eventually it will tell you, what you are currently looking at. So there's a lot of those. All right, so here's your data table, right? Similar to Module 2 labs. So let's go ahead and look at your next one, your enzyme lab. All right, so this lab is shorter than the other one. You only have 2 activities, and they go relatively quickly. So this is giving you background about enzymes. There's a really good video in the class announcements about enzymes and how they work. Here's the materials you're going to need, which should all, be in the kit. However, you are going to need hydrogen peroxide that's at 3%, as well as bottled water, and then some ice. So make sure you have those materials on hand. This is why I always recommend that you guys start these labs as quickly as you can, just because of the fact that you do have to go out and purchase items. So you don't want to wait till the last minute and find that you don't have the material that you need. All right, so here's your prep. Make sure you're reading through that really well and following it. In this first one, you're going to be looking at how concentrations of enzymes affect the rate of the reaction. So you're going to have the hydrogen peroxide, and you're going to be adding specific concentrations of it to the cups. And then you're going to look at how you have these filter discs, and you're going to be timing how long it takes the disc to rise to the surface of the peroxide. You're going to be conducting multiple trials of this, and then make sure you don't put your hands in it, like use the forceps to take everything out. And so then you have an equation that helps you calculate the rate of the reaction. So the distance that the disc traveled divided by the time it took to get there. Then you're going to plot it on a graph. Remember to use Google Sheets, and if you need to refer back to Lab 1, you can for help with the graphing activities. For activity 2, you're going to look at the effective temperature. Now, this is saying that if at least an hour has passed since activity 1, you're going to need to prepare a fresh solution, just because of the fact that it'll use up the materials and they won't be as effective. So you're going to have different temperatures that you're testing. Again, you're going to use the discs and time how long it takes for the disc to rise. So you're just going to see, okay, well, what's the difference between 15 degrees Celsius and 25 and 35 and 45, based on, how that disc moves. All right. And that calculation is the same. So like the distance over time. All right. So here's your data tables. Looking really quickly at that lab report, just because it's a little bit more detailed than the eukaryote lab. So you have your data tables here, and then you should have your graphs. You're going to have to copy and paste the graph from Google Sheets or whatever, Excel, whatever you're using, into the document here. Again, if you're not sure about how to do that, refer back to Lab 1 because it walks you through how to create graphs. All right, and then the third lab is the photosynthesis lab. So for this one, you're going to be um, looking at plant pigments from different materials. So this just kind of gives you the background about plants, which you should have already learned about because you're doing this in the next week. So you should have already read like chapters 7 and 8, which talks about photosynthesis and fermentation, light reactions, dark reactions. But there's also videos in the class announcements as well. So here's your background information, talking about the Calvin cycle, and then talking about chromatography. So you're going to need dish soap, you're going to need a coin, a light source. It's best if you have a light source that has like an incandescent bulb, but those are hard to come by, and in some places I think they're even like outlawed. So if all you have is the the LEDs, then you got to use what you have, right? But you definitely want a light source that gets warm. Um, and then you're going to want to buy some fresh spinach. You don't want to use old spinach because then you won't be able to see the plant pigments. So if you don't have spinach, you can use like kale or something like that. But once you are starting this lab, you want to make sure that you're cutting them correctly and you're using the chromatography paper correctly as well. So you have some chromatography paper, you're going to draw a line, and this is going to show you this video. Watch this. This is a really good video about putting the pigments onto the paper. And then you're going to basically just be putting a green line from the spinach. You're going to be rolling it onto the paper, and then you're going to have some isopropyl alcohol that's going to end up wicking up the chromatography paper and separating out the plant pigments so that you should be able to see different colors from the spinach leaves, like the spinach that you rolled out onto there. Then you're going to go into this next activity. Again, you're using the spinach leaves, and you're going to have the light, and this is where they're saying a 60-watt equivalent bulb is recommended. And then you're going to have a dark storage area. So you're basically looking at light reactions versus dark reactions. So you're going to cut out leaf discs from the spinach leaves, and then you're going to be putting them into water and looking at the gas that becomes extracted from them. This will make a lot of sense whenever you watch this video, so make sure you do that. And then you will be looking at the dark experiment. If you encounter anything that you're not sure of, just let me know, because I know that the photosynthesis lab can be complicated in understanding like what to do, but I can definitely walk you through anything that you might be struggling with.